So, good morning, all. Welcome back to our um, fourth week, third session, where we will be discussing about the art of the discussion. That is the discussion part of your report. In this case, we are talking about the report, and also when you are going to communicate it to any scientific publication journals, the discussion part need to be addressed. That discussion part can be a little bit different when you are looking from the um, discussion of a project where you have utilized AI. As we discussed in week one, that AI is more about generative and prediction. So we will be talking about the discussion part. Uh, so I hope that the voice is clear. You can hear the voice clearly, and I have also shared the PPT. Can you just confirm that the voice is clear, and you can also see the PPT? Can you hear me? Can you see the PPT? Hello. Okay, so we will go further discussing now here. The major part you need to understand with the discussion where the information is utilized from the AI platform. Uh, we are not interpreting the generative or the predictive. Why? Because we have used RAG, that is the retrieval augmenting generative AI, where the live data which is connected to the website, to the articles, are confirmed in our methodology and results, and that's why we are discussing the finding based on the existing literature and the data. So you need to understand that discussion part is very critical in the project where you are using AI, and it is important as you are part of the AI in predictive AI in clinical and pharmacovigilance. So the interpretation of AI generated evidences and the finding need to be within the existing scientific literature. And as a researcher, your role is very important when you're writing a discussion part. So based on the yesterday's result and the methodology and result part, you are moving into the discussion section. And why this section is important? Because the discussion is where you are actually transforming your observations, your raw finding, which you have mentioned in the results, into a scientific knowledge. So it actually, give a proper, layout to the research paper. Um, Result tells you what happened, but with the discussion, you are actually telling what actually matters, and what scientific context is important based on the results. So a well crafted discussion is very important because that gives the understanding, connectivity to the evidences that has been observed in the results, evidences for clinical purpose, for biology, for further research. So that is where actually the discussion come, and it is very important part of the any research communication. In this case, our report and for the article purpose, and where actually the common mistakes happen. The same we discuss in the result as well. Many times the student combines the result and discussion, and they continuously repeat the same thing what is there in the result, but discussion goes beyond the data. It has to be interpreted by connecting with the citations. So there will be claims based on the support. You cannot put the claims and you are not supporting it with the articles. It is going to reduce your claims argument significantly. Then you are selectively reporting only the supportive finding, is like a bias there. You need to make sure that you acknowledge or address if there are any disagreement in the literature. So many time what happens, the result section when it is being written down, um, and then you go for discussion, and in discussion you don't have any literature to support. At that time you need to make sure that you are also looking at the articles which are actually disagreeing with your finding, because the same thing you can cite, and you can also make sure that you can acknowledge that the work which has been similar work which has been done previously is actually disagreeing what we have done. So that need to be make sure you have been putting in the discussion part. Then you, in a measure of the time, you start concluding that section. So the conclusion section will be there, and you need to make sure that the discussion section should be only for the interpretation purpose. You shouldn't draw any conclusion in the discussion section. Whatever you have observed in the results should be interpreted in the discussion. Then one of the major mistakes that we have found in the project, in the research which Swalife is doing with the intern, is that copying AI output directly into the discussion section. So AI generated text you should be avoiding now. When we are saying this, you are going to use AI as an assistant, but you need to make sure that what you should have already had some interpretation in your mind, and based on that you are actually designing your prompt, so that way the result uh, which you have got with the help of LLM, you can actually write the discussion sections. That is important part. That we are not saying that you shouldn't use the large language model, but the large language model should only be used as an assistant in a writing section, not the output of your LLM should be included in the discussion as it is. Otherwise, it is not going to be, the output which has been given by the LLM can be generative or can be predictive one, and it is not going to be the one which is interpreted based on the result that you have got. So if we look at the why it matters with when we are using AI. So in traditional research, manual review was done, extraction was done manually. So data evidences was collected by hand from different sources, which was limited based on the access, based on the limitation of the researcher. And then manual synthesis of that finding was difficult, because you have to utilize manual process, and then you are putting your judgment and then drawing the conclusion. In this case also you have to do, but the previous all the procedures are now utilizing AI. So a RAG retrieval method, when you use, you are actually touching the databases relevant to the topic that you are doing. So the literature will be collected using the retrieval augmenting generation model. That is the in your case you have used perplexity, and perplexity is always connecting all the information with the literature. But again, your role as a confirming that the articles, the information is there with with your judgment is important. Then AI is extracting evidences, biomarkers, and target, and that's why the scientific prompts are there, which help extracting all that. And then you are organizing them, summarizing them, uh, with again utilizing the AI. The prompts will be specific for your data. Now, if you look at the predictive intelligence, clinical, or pharmacovigilance, we have the prompts for the artifact purpose, that is for the data purpose. But the next sections, that is the result and the discussion part, has to be tailored to the information that you got in your in your raw data. And that's why at the end, scientific judgment remain with the researcher, and based on that you are actually interpreting that. So that is where actually the change has occurred. So if you look at the traditional research, coming to the scientific judgment with the information was taking years. Even with the when you are submitting an article to a journal, and it getting published will take minimum like 1.5 year, 2 years. With the AI now, that is reduced, and also the information, the data which you have got from wet lab can also be put into the format faster. So, what does the AI actually produce? So, you need to understand the output of AI research tools are essential when we are interpreting the discussion section. So, literature summary, which are condensed into relevant published article. Then you will also have evidence tables. So, these are major of the part of your your results. And then when you are discussing them, you need to make sure that you are picking the relevant information that you have put in the objective. So literature summary, evidence tables, biomarker extraction, then target identification, safety evidence summary, and clinical evidence summary will be part of your result, and also your discussion section based on your objective. So, for example, in the predictive, if you are saying that TNF alpha is, you will be focusing as a target, and in your result you have observed that many of the studies which have shown in the literature are targeting TNF alpha in oral cancer. Let's say we will take oral cancer only. So that information now, when you are discussing, you need to make sure that you are connecting the studies which has been observed in your result in the discussion, saying that why TNF alpha was focused in this study, and simultaneously, uh, why the patient with oral cancer, or which type of oral cancer patient can be treated with the drug which if you're working with should target TNF alpha. So similar to clinical, you will have similar to safety, that is for pharmacovigilance, you will have adverse events or safety signal relevant the molecules or the combination in that specific disease. So you will have that information which you observe as a result, and then in the discussion section you will be adding the with the literature, supporting literature, the interpretation. So I included this slide to just to clarify that when you are putting any prompt in any of the LLMs, majorly in the perplexity, if you select the models from Gemini, um, Cloud, or ChatGPT, they will majorly go for predictive and generative. But if you select the perplexity sonar, or if you connect website in cloud, ChatGPT, or in Gemini, There you will have evidences, because in the evidences the information is extracted from the published literature. So if you look at the the statement in the evidence, 18 studies reported TFKB involvement in the uh, FKB involvement in the inflammatory disease progression. So this sentence tells you that the 18 studies were there. Now, in these 18 studies will actually have the citations. But when we look at the prediction, it says that patients with elevated NFKB may have higher disease progression risk. So the sentence are similar. Uh, the target, that is NFKB, is a major target in the progression of the disease, is well established role, but that is a prediction, whereas in evidences it is supporting with the article. So you need to make sure that when you are putting information in result and discussion, you are putting the evidences and not the prediction. And when the content which you got from the large language models are without the references, it will be the prediction or the generative one. And there where your role is important, because you are looking at the information from the evidence point of view and not from the uh, generative and predictive point of view. So the workflow wise, if we look what you have done in your project and what need to be done, is that the research question was given to you, which with the help of the scientific prompt and the large language model, you have got literature retrievals and the RAG systems, which is giving you the evidences to extract, and those evidences that you have extracted need to be validated. Now validation will be always done by the researcher, by the the scientist who is working on that project, the research. So that is where actually your role as a doing this project comes, because the content which you have got from the large language model is connected to the live citation, but that need to be confirmed, that need to be located, the original publication. Then reading need to be done regarding the information that you have got from the large language model, and need to confirm that information. That is the evidence validation. And when you are doing this, that will be the you know in result you observe, that is you got the evidences that you extracted. Now you validating them, interpreting them in the discussion part. So you need to make sure that the evidence has been verified in your discussion part, and then you will come for the conclusion. Now when you are doing the conclusion, you need to make sure that the conclusion shouldn't be in the discussion part, that we have also previously talked about. So where actually your discussion began, it is very important. Many of what happened, many of the journals actually have result and discussion as a one part, and that's why sometimes as a initial researcher we get confused that uh, when I'm writing result and discussion together, where actually is the discussion part and where is the result part. But in many journals and also in our case, we are keeping discussion separately. So discussion is the interpretation. So once you, the first step in the discussion, once you have got all the finding that has been documented in the result section, you got a clear presentation of information that you have observed, and that is in the result. Now you verified that evidences. So uh, the output of your large language model uh, that you have included in the result will be viewed and validated against the the literature, the source which is available as a original in the form of original articles, reviews. and then you are going to connect them with the key themes, with the patterns, because in the result there will be numbers, there will be graph, there will be patterns, and also any contra indication contradictions have been identified that you will be interpreting in the discussion section as a validation part, and it will be structured as a discussion, and only then only your discussion begins and it will be completed once you verified and organize the evidences that has been observed in the form of patterns, in the form of the graphs tables which are there in the result. So the interpretation actually is done and verified with the organized way. So this is the framework for writing the discussion. So discussion section should ensure that the major key element which are observed in the results are addressed in a systematic manner. So you summarize the key finding. So you open with a concise synthesis of the most important result based on your objective that you have included in the introduction. Then you contextualize the finding against what is available in the public domain in a published format that can be supporting or that can be actually contraindicating your findings. So you need to compare that literature, articulate the mechanistic and clinical relevance of the finding. Now, again, when you are explaining that significant observation that you have included in the result, you need to make sure that you are articulating in a way that it is not stopping the result there, the discussion there, the interpretation there, but it is helping to make sure that a further decision regarding the finding can be taken. So that way, the further work can be done. And you also need to make sure that the acknowledgement about where your study limitations are. Now, in the publication, not in the report, complete, everything will be done. There will be limitations. So, let's say, for in our case, we are not doing wet lab. So, you can actually, tell that limitation that it was a virtual AI-based studies utilizing the large language models and the scientific prompt. um, you are having limitation with wet lab. So if your study need to be performed for the wet lab or can be taken into clinical, you can actually mention that as a further um, for the limitation and acknowledging it where you can take as a further studies. And then the last is the future research that I have told you. Once you got the limitations, you can have a directions where the further studies can go, but simultaneously you will have a gap as well. So whenever some initial research is starting with any group or when any student in case of masters or PhD, they're entering in the field where they are going to continue in the field for many years if they are going to stick to the research. At that time, it is very important that when you are writing your any scientific research, you acknowledge the limitation of the work, and you also suggest the future research because you have identified the gap, and also you propose the further directions which you are going to go in the future investigation. So let's look, this framework step by step, that what actually you are summarizing from the results. So when so we earlier we took one protein, but in this key finding, let's say in your result you have identified that many of the studies who have been working with TNF alpha. So specifically when you are in the result you are specifying the number, so 30 studies were actually you know um, given by the large language model. Out of that you have identified 22 who are talking about TNF alpha. So in your analysis you included those 22. So that is where the result comes. Now in the discussion, you are going to say that TNF alpha is, becoming one of the emerging target as a inflammatory. If the 22 actually is talking about inflammatory, you can talk about inflammatory. The 22 is about the disease, specific disease. In our case, we have oral chemotherapy and oral chemo prevention. So if we have identified, let's say, 3 4 articles, and out of that 2 articles are talking about TNF alpha in chemo prevention, that gives a strong association because there are only 4 5 studies. Now that can be limitation as well. You can also have a further studies where you can focus on TNF alpha and do a similar type of study, taking a specific samples of the patient and also from the preclinical. So in the discussion, you based on what you have identified in the result, you can actually interpret it as a TNF alpha as a major target which can be associated with that specific disease, because out of 30 is 22, that is about 73% study has included that in the study. So that is the interpretation. So the key shift is from what was found to what it means. So found was TNF alpha in 22 study. What it means is that TNF alpha plays important role in that disease, and it can be a major association between that disease. So that is where the major finding you can summarize clearly and concisely in your report, in your article that you want to communicate. Now you can also do comparing findings which are there in the literature. So let's say after doing the literature studies, you have identified studies that collaborate with the results. So there are citation from specific authors, years, and also some of the finding are relevant. Then there are also some information about specific like age or categories. Maybe a patient from similar category of disease you have identified, or you have found that there is a consistency across the context, like considering whether the agreement hold uh, across different population or same population with similar methodology and also similar disease setting. So, for example, in here we are actually done a literature, and there they have identified that the biomarkers, that is TNF alpha, the protein, have shown majorly in 22 studies. Now, in that, 18 studies are supporting, 18 studies are actually having a contradictory studies. So those 22 studies are actually showing that TNF alpha was used in the study. 18 studies are supporting, so there is a strong evidence. Four are actually against. Then there will be another biomarkers like NFKB, where they are utilizing pathway activation, and out of 30, 18 has actually supporting, where three are uh, so out of 18, 15 are supporting and 3 are having a contradictory information. So that comparison is actually looked in your result, and you are making sure that you interpret. So in the result you can say that TNF alpha is actually in 22 study have shown promising, but when you are putting in the discussion, you are interpreting that out of this 18 study was strongly supporting, that is about 73% are actually, and that's why TNF alpha can be a major biomarker in this study. So that comes in the in the discussion, and that is comparison from the literature. Now, in the third step, you are going to explain the biological relevance, where if you take the same example of TNF alpha, a master regulator of inflammation. Why? Because 18 study, that is about 73 about from the 22, are actually focusing on the inflammation, and they are positive to show that TNF alpha was the major role player in the inflammation. So There is a consistency in the evolution which suggests that TNF alpha plays important role, and that's why you are explaining the biological relevance. Same thing with NFKB, then interleukin 6, and also for VEGF, if you have identified. Uh, why I'm taking this, because with the cancer, with the oral cancers, we have identified these target biomarkers in the studies that we are doing. And in step 4, if there are no information available, but there are contradictory information available about your research, you need to make sure that you engage with those finding, and you should actually include them, you know, with the discussion, where you are informing that this negative uh, or this information are there which are contradictory to your finding. So you're not going to dismiss those findings, you are going to have that finding as part of the discussion, because again, uh, the way it works is that your observations in the results and putting them in discussion will be based on what you have got the information about the area. So sometime, you might actually find just one study which is talking about positive, and then when you are actually doing the similar type of study, you also are in the positive state now, because that study, that one was talking positive, you have validated that. You will have many negative studies which are against that one, but you still have to include all of those information, so that way when you are taking it further in the future studies, the gap will be there, and your further guideline for the research will be there. So you should always make sure not only the positive studies but also the negative studies should be there, and you should actually make sure that the discussion is going to acknowledge agreements and the disagreements, so that way you can actually strengthen your paper strongly. So study with partial or contextual dependent results should also be included in the information, so that way you can actually make sure that your scientific argument becomes stronger. Then identifying the known gap will happen when you actually make sure uh, when you make sure that you have mapped the articles relevant to your observation, that is relevant to your result, and the current information that is available, because identifying the gap is actually important part. It's not just a uh, you know um, so you are basically contributing the scientific committee. It is not about failure to admit the scientific gap. So as I told you earlier, that limitation of your studies, limitation of any scientific evidences, limitation of any preclinical evidence, because the sample size was less, or the study was conducted in that period which need to be longer, or insufficient mechanism studies was not done. Those are important gaps in your study, and those need to be addressed in the discussion. There will be always limitation. When you acknowledge them, then only actually it gives the power to your scientific judgment that you have done in the paper. You cannot just continuously defend what you have not done and not included in the paper. So let's now we will talk about the study wise, predictive intelligence, clinical, and PV wise, that what discussion part should be there. So in case of the predictive intelligence, the lot of information that you are going to get will be about biomarkers, co-occurrence, clinical, which is going to come across through different literature. You need to observe and report them in the result in a structural format, and based on that you will come to the interpretation that will be in the structural format regarding the biomarkers, regarding the preclinical, clinical studies, and the association of those targets leads with the disease. So you are going to have a clear picture about identified target and the therapeutic potential of the molecules or the leads that has been allocated to you. So that is going to help us rank the evidence strength of those molecules in the research, basic research, clinical, preclinical, clinical, and also those molecules, because they are from plant, small molecules. So there will be also have some product which are in the market. So that is going to strengthen the evidence and will have a strong interpretation there, the output there, which will be involving confirming the biological plausibility of the identified target biomarkers. It will also gives you frequency against the broad literature, and also articulating the potential need in the therapeutic target or for potential for the diagnostic purpose. So that is where the predictive intelligence discussion will lead, and that is what you need to include in your report. So targets and the pathways will be there, as I told you. So for example, in curcumin case, curcumin is the lead you have identified, and it will give you all the targets and the mechanism which they are focusing when they target those proteins. So when you are actually got the observation that is in the result, you need to make sure that when you are discussing, you are focusing on the target finding, explaining not only which target were identified, but also how they interact with the disease. That will be also information will be there. What evidences, basic evidences, preclinical, clinical evidences, and the studies which have actually utilized that target, uh, how there is a modulating process going on in the target when they are being utilized in specific disease studies. In our case, we are talking about oral chemoprevention, so there will be connection between those target and the pathways with the supported evidences. So that is what you are going to have as an interpretation in your report. So you are translating the information. Translational research is happening here. You are bridging the gap of the finding which you are going to get from the large language model, which can lead. I'm continuously telling that you will lead it towards the preclinical or clinical studies. Our aim is that we will have enough evidences that the molecule will be leaded towards the clinical purpose with the known biomarker that we want to focus with those molecules that we have identified. So there will be prevention. Simultaneously, we'll also have finding for diagnostic purpose, and then for the precision treatment, where a specific target will be identified, which will be a actionable therapeutic opportunity there, which can further put into analog or into formulation for that specific disease. So that is where the predictive intelligence. Now, for clinical research purpose, the discussion is mainly about the clinical trial summary, about the formulation and the disease that we have selected. So you will be talking about what clinical trial you have got and the summary which has been given in your result. The outcome of those report, the primary, secondary endpoint that you are going to include your clinical trial protocol. You will also have some primary and secondary endpoint in the clinical trial that you have identified from the databases that you have put in the result, and you need to now discuss what outcomes were there, similar to what actually happens in the systematic review that we discussed last week. So that will go there, and that way you will have a information. If there are no clinical trial available, certainly there will be some indirect clinical trial which will be given by the databases that you will be including. Then the endpoint analysis, the statistical, the analysis part of that specific, in your case, it's a cross-sectional study. So you can actually talk about cross-sectional analysis, but if there is a study available, but the studies which has been identified by the databases, you are going to talk about those endpoint where the overall survival, the progression free survival or quality of life of those patients in that clinical trial, what was actually reported by the clinicians. So discussing clinical significant is important part here, which is going to actually discuss and the difference between statistical significant and clinical significant can be identified. So these are not the same thing, but majorly we actually try to correlate it, but in statistical. So let's say example, when a finding is statistical significant, when the probability of observing it by chance is below a defined threshold. Majorly, if you see p less than 0.05 is the value. Now major studies like 0.005, 0.005 are being reported. So determined by the sample size and variance, a large trial can make a tiny difference statistically significant. Does not tells you whether the difference matter in practice. So many times the statistical significance doesn't mean that uh, the trial actually have given us the right information. So if you take an example, a drug reduced blood pressure by 1 mmHg with P is equal to 0.001 in a trial of 50,000 patients. Now, when the p value is equal to 0.005, that means it is a statistically significant. But if you look, it is actually just showing the blood pressure reduced by just one, and that is in 50,000 patients. So it is statistically significant, but it is not clinically. Because in clinical significance, if we take an example, a drug reduced blood pressure by 15 mmHg, a difference that meaningfully reduce the cardiac risk. In the patients, the number can be 50 or 100. Now the reduction was 50, so it is not clinically significant. It is not statistically significant, but it is actually giving you clinical significance because the magnitude of the effect is large enough to actually have impact on the patient. So that is important when you are actually identify the trials. Make sure that this information is highlighted in the report. You need to make sure you determine the effect size and the clinical context, require expert judgment about what constitute a meaningful difference, which will be there in the clinical trial, and guides real world treatment decisions. So based on that, real world treatment decision is going to be taken. So you need to make sure that when we are talking about the molecules which we are working on, if there are study available, what actually it means, you need to come with that information so real world decision can be taken. So with the clinical finding, statistical and clinical significant, the discussion must address whether they can be applied just beyond the study population. So that is very important. You need to understand the different population where the trial participant represented. So when you actually have all those trial, you look them from that angle. You are going to see the age of the groups. You are going to see the health setup, and that is in the clinical trials which has been identified by you in the databases. Now, with the pharmacovigilance, discussion requires careful and evidence-based approach, where the sources are going to be providing you safety evidences regarding the molecules, the allopathic drug, and the disease. And based on that, you will have case reports of different patients or any adverse events which has been occurred, and those are on social media or from different databases. So valuable hypothesis generation. Uh, need to be there, which is not only going to be established based on just what you got, but simultaneously from the discussion it will be created. So all the case studies that you have got and put into result, you need to make sure that you include in the discussion as a interpretation of those case study that will be then useful to establish the causality or even the hypothesis. Then, like I told you yesterday about in the methodology, you need to include day before that. I think you need to include the databases. So any finding that you have got from the databases, it can be any databases, need to be included in the result and then included as a interpretation in the discussion section. So any signal that you have detected in the result will be interpreted, and any underreporting regarding that will be identified. So that interpretation need to be done, and it has to be done precautionally, because sometimes maybe like different databases. So if VGBase is showing something, and if there is no FDA adverse event reporting system have that, so you need to report them precautionally, but you need to still highlight that this is what is being given by the VGBase or by the FDA adverse event reporting system. Then the literature evidences. So you got the database separately, you got the literature evidences as well. Any published studies like case reports, then all the the observational studies, case studies, systematic reviews are there. Then pilot, feasible. So all those clinical studies are going to report if there are any adverse event which has been occurred in the studies that was accessed by the large language model, that need to be included in the report as a observation and then need to be discussed in the discussion part. Now connecting the regulatory to that, because based on what you have identified, also what actually information is available from the regulatory point of view, that can be prescribing information, risk management plans, and the communications of the regulatory agencies should be included as a discussion part, representing the official position regarding the molecules, the formulation that you have been working on, if there are any safety concern. Now, with the molecules which we are working, maybe you might not find in any of this, but still there will be information from literature, as many literature, many studies have been performed with those molecules, and you will find something based on that. But again, as I told you, the model of perplexity is also going, if there is nothing, it is going to give you some of the prediction of the information which can be included in the result and the discussion part regarding the molecules which we are working. So when you are establishing the risk or the signals, you need to actually make sure you uh, use a language which is describing the safety concerns, precisely reflecting the strength of the data that you have got in the large language model. So if you actually make sure that the terminologies which you are using is being used proportionally, then there will be no unnecessary or like under statement of the risk or high statements of the risk. So you need to make sure you are using like potential signals. So if there is a early signals which are difficult to confirm, or if there is any difficulty in actually creating association between those signals, then you need to make sure that those are reported in a way that there is a limited case reports or preliminary data available, and that need to be communicated in a proper terminologies and a proper use of the language. Then if there are emerging signals, that you have identified in your results from the information that you got in the large language model from the databases. You need to make sure that the pattern that you have identified uh, is going to be, need to be in the um, consistent manner, so that way you can actually report a strong association. So when I'm saying this, like I told you in the previous slide, that if you have identified a signal in FDA reporting system, and same signal has been identified in VGBase or in the articles, you can actually connect them, and it can give a strength. It can give a strong signal, and can be, utilized as a emerging signal relevant to that molecules, to that formulation in that combination for that disease. So that is where, emerging signals, and the confirmation is like a supporting association between many of the databases for that, from all the databases they are supporting. At that time, it is going to be a confirmed safety signal. Now in your case, because we are working with the small molecules from plant, having signal, a potential signal, is also very difficult, because major of the time the person is taking, the allopathic and the molecule together, and once they stop, that potential signal is stopping, so they are not reporting that. So major of the time we are having difficulty to get any of the signals from the databases, and that's why any information from the published literature is going to give us early sign of the signal, so that you need to include in your discussion. But when you are including in the discussion, you need to make sure that what is signal and what is the established risk. Established risk is when actually you have got signals from the databases and also from the literature, which is, given by multiple studies, and regulatory have reviewed those information. So many of the plant also, the regulatory tells that don't take these plant tablets or capsules in combination with certain drugs. So that is where actually a strong established risk has been already confirmed. Then, in addition to pharmacovigilance, you will also have information from clinical and regulatory point of view, which need to be there in your results and which will be included in the discussion. So, does the evidence actually justify the monitoring? Is a need for change in the label? Is there any additional research required? All those gaps are going to be identified, and that's where actually Life is working, because we are doing pharmacovigilance from the research point of view, from the preclinical point of view, to understand if there is a major gap I will there for the evidence purpose for those small molecules, which cannot actually, give you the definite conclusion. Then again, whatever is available in the public domain regarding those molecules, in the formulation point of view, which has been created by different companies. So evidences are there, are evidences that actually can utilize for further research, for understanding the label, which need to be, showing adequate information for the different countries. So that is there, and is there a potential monitoring need to be done for those formulations. So uh, that information you are going to get, which will be in the result section, you will be including, and that in the discussion you will be adding. So now I had one question, after, couple of sessions that we have, uh, that should we actually use AI in the writing, when we are writing the results methodology. So yes, means I have replied to that question, but I included the slide here because, it is based on what you are doing with that AI. You need to make sure that you are utilizing it proportionally. So AI can actually help you draft the text, summarize the evidences, improve the language, organize the arguments. Why? Because it is majorly the generative and predictive models. You, when you are working with the research, scientific research, the evidences which you are going to get, you cannot validate them using the large language models. You cannot come to the conclusion because you are the one who are going to come to the conclusion. The conclusion can be right or wrong, but you are not deciding on that. You are coming to the conclusion. So that cannot be done with the AI. And the scientific judgment, that is, not you are enough evidence is available. That scientific judgment LLM cannot give. You can try it. You can tell the LLM that you are a researcher, and based on your expertise, can you come for scientific judgment? It will come on scientific judgment, but based on the domain expert, what you have learned, what you got, you are the one who will take it, the information further by doing a critical thinking about it, and then making the discussion which is going to be useful for the scientific community. So make sure that when you are including AI to write your methodology, your results, your discussion, they are going to draft the text which can be right or wrong. If you don't do that judgment, it is not going to be useful as a space for the scientific community because it is just generated from the from the large language model. So you have to remember this. So the key takeaway from this session is majorly the six principles, that is AI generated evidence summaries, not actually the final conclusion. Discussion is where the researcher adds the scientific value. Then every discussion point must be linked to the evidence, that is to the results. Contradictory finding need to be included because that actually strengthen your scientific rigorness. So that way you are making sure that you are including honestly if there are any disagreement in the literature in your work. You are going to talk about the limitation which are part of your studies, and you are going to acknowledge them, that what limitations are there. And you are going to use AI for writing assistant, but you need to make sure that the critical scientific decisions about the you know methodologies, results, then the discussion has to be taken by the author because the accountability is to the author. The content of the discussion is part of the author. So that's about the discussion part, which we have discussed from the AI point of view as you are utilizing the large language model. So what need to be included in the discussion and how we have discussed that. If you have any question, I can answer them. Tomorrow we will be talking about the general, the high impact abstract titles and the keywords which you need to include in the report and also for the publication purpose. Okay, so if you have any question, you can ask or you can put that in the comment box. Okay, so I will see you tomorrow at 11 a.m. See you.